Syndrome. So a very good evening again to all the participants. So this topic of today's is naming. I just left it out that much because it is about publishing new fungal names. So publishing new fungal species. This is all about publishing new fungal species. So when you have to publish new fungal species, there are some codes, there are some guidelines which we all need to follow. So there are some checklists for publishing the new fungal species. Now those checklists are very much important, and among that checklist, what you have to follow and what is of significance, they are of, we will slowly proceed as we are trying to understand what it is. So this area is something that is even difficult to understand, and it will be new for, including myself, for many of us it is new. So why this is important? Because over the period of time, we have seen several new fungi, many species being identified, and they are named, and we find several names being provided to them. And we have Canada Vishwanathi, we have Malassezia arunloke, we have Cunninghamella arunloke, then we have the Madurella mystromatis, as we have got Madurella fahali. So there are a lot of names which are after great scientists, also great personalities in mycology. So we are trying to understand publishing new species. What does it mean? So there is a code. So if you want to understand this, first you have to know what is a code. So the International Commission on the Taxonomy of Fungi, that is ICTF, they first published a paper which designed to give guidance and provide best practices for authors for proposing new names. So it was long back around 1987. So this was somewhere around 1987. So later on, there was a code that is very important. So what do you mean by this code? So code is actually, it stabilizes the usage of names by international agreement. So first they have to be internationally, it has to be agreed by a set of rules. And what are these rules? This is guided by a highly technical document which is called as a code. So a general code, a general account of the code is now published regularly, that is known as the code decoded. So these are some of the important aspects when you're trying to cover for the publishing new names. Now, in each code, actually, it becomes obsolete when you have another fresh code coming up. So each previous code becomes obsolete. Each previous chapter becomes obsolete. So there are new chapters which are added on with each and every new code or new meeting that comes up. So new provisions in the code are also retroactive unless specifically limited by the data or the date. So provisions specific to fungi have been separated into chapter F, and this chapter F is coming under the ICN. Okay, so this ICN we have understood it yesterday. So ICN AFP, AF is for the algae, fungi and parasites. So this is algae, fungi and the parasitoids. So this is how it functions. So what does this chapter mention or recommend? It recommends for the sanctioning, protection via lists of names, registration of names and typification acts. So we'll slowly understand what is this. There will be a sanctioning, then there will be protection based on the list that these names are already registered. These names are already, the list of names are already existing, and these are some names which are proposed. So we register those names, they say, and then there are typification acts, whether this is a holotype, whether this is a epitype, whether this is a new neotype. So there are different things that happen with this naming procedure. Now, what are the relevant changes made to the code? So there have been relevant changes that have been made to the code since 2010, when the first publication of Seifert and Rossmann was done in 2010. So after that, actually, there have been a lot of changes what have happened with the code, which we already know because we have understood taxonomy so far. So elimination of dual nomenclature, which has been actually making the nomenclature easy. Elimination of a Latin requirement. So we know what do you mean by dual nomenclature. There was a teleomorphic name and there was an anamorphic name. So this was always confusing us. So this dual nomenclature has been removed, and it is no longer acceptable to apply two or more scientific names for a single fungus. So it is not acceptable. Now it is, earlier it was like a necessity. Now it is such a thing, you cannot have two names for same fungus. So that has been one of the very important aspect of elimination of the dual nomenclature. Then the elimination of a Latin requirement. So when we have the binomial nomenclature system of Linnaeus, we have been insisting on the Latin requirement. So this elimination of Latin requirement is again something which is very important in the change that has occurred since 2010. So Latin is no longer required for descriptions and or diagnosis, and this can now be either in English or Latin, but not in any other language. So it is not mandatory that every name has to be Latin, but you can have an option of English. But again, does not encourage any other language. So this is under Article 39.2 of the code, what is mentioned. Then you have got the registration of the nomenclatural novelties. This is again very, very important. So whatever new you are trying to, novelty that you are having, so it has to be registered under the nomenclatural bodies. So this aspect is again very, very important. So these nomenclatural novelties include either new taxa, replacement of an old name with a new name, or a new combination, what you want to take up. So this must be accompanied by citation. So you need to have a citation and identifier number, which is obtained from a repository that has been appointed. This repository would have been appointed, which is usually called as the Nomenclature Committee of Fungi. And this again is ratified by IMC. So every new published, what you are trying to name that has been published, every new name that is published, it must have a separate identifier. So this is very, very important. And with every new genus name that is published or with one or more new species name that are being published, there will be separate identifiers. So what we require is this registration of nomenclatural bodies. It is actually stepwise, and you require several identifiers. So this has been discussed much detailed in Article F.5.1 of the code. So at the time of writing, what you have to do is the recognized repositories, like we were discussing about the Mycobank, the Index Fungorum. Okay, so these are some things which are very important, or the fungal names, what we have submitted in the fungal lingo F.net. Or these are some of the components or some of the repositories, recognized repositories where you are submitting. So they give you identifiers. So it is only necessary to obtain a single identifier from one novelty. So you cannot have multiple identifiers by submitting to multiple repository banks. The standard method of citing the registration identifier is to place the number after the name of the relevant repository. So you give that number. For suppose you are, which repository you are placing it in, like FN for the fungal names repository, Index Fungorum, you write it IF. If you are putting it in the Mycobank, you write MB after that identifier. And it is separated by a space. So that is how usually it is written. Next very important thing is registration of new typification acts. So this has been, this has started actually as on 1st January 2019. Any new type designation such as lectotypification, neotypification, or epitypification of the previously existing taxa or at or below the rank of species, they must be registered. And the identifier must be cited with the typification. So you have to, actually it has to be registered with the typification. So this comes under Article F.5.4. So what they are asking us here is, every kind of, we have to describe first what is the typification that we have chosen, whether it is a lecto, neo, or epitypification, which we will slowly understand what it means. So this every kind of typification from the existing or a new one, you have to register it. Even if you are changing an old name, it has to be registered under this registration of typification acts. Then there are types of cultured fungi. So as of 1st January 2019, where a type is derived from a living culture. So the method of preservation must be clearly indicated as being metabolically active. So you have to tell that this type of culture, what is the nature of that culture, and if it is metabolically active. This has to be accepted as the type. If it is metabolically not active, then there will be issues. So you have to tell that this is preserved and this is metabolically active. So this actually culture preservation, what you have done, and it has mentioned in somewhere in the code in the article 40.8. Now, cross-code homonyms, that is again very important aspect which are there. Like homonyms are names of the families, gender or species spelled exactly the same. Suppose there is some name which is already existing, you are giving the same, you are proposing the same name. So that is cross-code homonyms. So that is again checked. So it is a name, if a name is already existing, so it becomes illegitimate. So you cannot use a second name of similar kind or similar sounding or similar spelling. So that is called as the homonym. So from 1st January 2019, the names of the fungi must not only avoid being homonyms, but of names of other organisms covered under the code, but also it should not be a repeating prokaryotic or a protozoan name. So this is very, very important. It should not be a protozoan or a prokaryotic name. At the same time, it should not, you should have to avoid the homonym. So now we have a lot of names that are changing. No? For Penicillium marnopi, we have Talromyces marnopi. Now at the same time, we have understood that there was Nemocystis girovaceae. Earlier it was called as Nemocystis carini. So these are changes in the names, and you have to address to these cross-code homonyms and why it is being done, and is it legitimate or illegitimate that you have chosen. So these are some of the important aspects of the code. Now, the code decoded. What do you mean by the code decoded? Once you're trying to decode the code, so you are going around for publication. So effective publication. So there are terminologies of the code decoded. These three terminologies cannot be interchanged or misinterpreted. First terminology is effective publication. Second is valid publication. And third is legitimate publication. Now, these three actually have a different meaning. So in total, so the effective publication term has been described in the articles 29 to 31. And effective publication, it concerns the kinds of publication in which names of new taxa can be published. So this after January 2012, effective publication can include publishing in online only formats and must adhere to certain requirements, including that the online document is available in PDF and has an international standard serial number, that is ISSN, or an international standard book number, that is ISBN, or explained, that has been explained very clearly in the Terland. Terland is the guideline where you are reading about how the code is decoded. So for effective publication, it is very, very important that now after 2012, January 1st, 2012, only online only formats are prescribed. Then these have to be in the PDF format. They should have ISSN number, that is the international standard serial number, or international standard book number. Then they have to, as it has been explained in the guideline. So when publishing online, they usually discourage placing nomenclature novelties and new typification in supplementary material, as these are not readily discoverable. So you cannot put anything in the supplement. It has to be in the main one. So only supplementary material is not effectively published unless it is in the PDF format. So this is all about the effective publication. For new names, what is the effective publication style? Then valid publication, what is a valid publication? It is dealt with in the articles 32 to 45. So for a name of a new taxon to be validly published, we are talking about validly published, it must meet the requirement for effective publication. First, it should meet the requirement of effective publication and meet the additional provisions laid out for valid publication in the current codes. The codes, whichever are current, you have to follow that. So these include provisions for correct designation of holotypes or basonyms and registration of the names. So if you have holotypes or basonyms, you are trying to understand that and you have to register the name. So if the published name, what you are saying is not valid, and such a thing will be omitted, because you know that there is no identifier for such a name. So it will be definitely be omitted. So that is, so close attention has to be required for achieving the validity of the publication. So valid publication is something that is very, very important. Then there is a term called as the legitimate publication. So in addition to meeting the requirements for both the effective as well as valid publication, a new name must be legitimate in according with the rules of Article 6.5. So illegitimate names include homonyms, that is names spelled exactly the same as the previously published name, or a new name that includes the type of a previously published available name or epithet that should have been adopted. So these are some of the very important aspects of the code decoded. And when we are publishing for the new names, it has to be effective publication, valid publication. Effective is the online mode, ISSN, ISBN, that is ISBN, and it has to be in PDF format. Supplements should not be in, it should not be a supplement, but rather it should be in the main format. And valid publication is what we have known, that it should have an identifier. And second thing is, it should not have any holotype representation of basonemes. Legitimate publication is something that again is a new name. It should not be a homonym. It should not be homonym. So designation of a type, it actually serves as a physical reference for a name. So the very important aspect for valid publication is again typification. So we'll try to understand what are the various designations of a type. A designation of a type which serves as a physical reference include the holotype, the isotypes, the epitypes, lectotypes, and the neotypes. So new species actually must include the designation of a holotype setting or holotype specimen, and this must be deposited in a single specified institution. If more than one fungarium or herbarium or culture collection is designated for the holotype, the name becomes invalid. So you should have only one center or one institute where you are submitting that. So this becomes very important. You can have multiple fungariums or multiple places where you are herbariums or the culture collection centers, like we have NCCPF. Suppose that one culture collection we have submitted it, that becomes a holotype. Now we cannot go ahead with submission with another culture collection. So that becomes again invalid now, because you lose the characteristic or the property of the holotype. Now isotypes, they are genuine duplicates of the holotype, and that may be deposited in the same repository that holds the holotype, or preferably in additional repositories. So these are isotypes. So when you are having one holotype which has been identified, now you want similar isotopes, similar organisms you are getting, and now you want to expand it to further repositories or further culture collection centers. So what you are going to do is, you will mention it as a isotype. You will not write it as a holotype, because holotype is already existing somewhere. So you have to mention this isotype is correlating with which holotype, and that holotype is submitted where. So this isotype can be submitted to the other repository or other culture collection. Now there are something called as the epitypes. So epitypes may be designated and attached to any existing type when the original type material lacks sufficient characteristics or that are unambiguously identified. When designating an epitype, a reference must be made to the type that it supports. So you already have a holotype, but there are some issues or there is some anonymity with the identification of that particular holotype. So you are getting the epitype here. So this epitype, it can be lectotype, it can be neotypes. So these are further types. So types, when we say in case of new names, when you're trying to discover, so we have got so many types that we need to understand. So these are some things which are very, very important when we are trying to describe the new species. So this International Code of Nomenclature of Prokaryotes, it does not require that the description should conform to any minimal standards for code compliant publication. So that is again very, very important aspect. So the goal, we are reading about the taxonomy. Taxonomy has its own significance. It has got a lot of importance. That is why we were stressing in our first lecture that there have been changes in the names, and we need to accustom ourselves with the new names of the fungi. Because if we do not remember, if there is something new, what new name you are going to give? If it is a repetition, so we have to have an updated taxonomical aspect with this. So the taxonomy has some goals. It provides all necessary data for both the contemporary and future workers to identify the taxa and use associated data in the downstream analysis. And it also reduces the likelihood of publication of taxonomically superfluous names. So those names which do not exist or which were wrong, so you don't keep on publishing those names, or you don't keep on writing about those names. So this is very, very important. Then when you're trying to write a new taxon, there are many things that you have to refer. You should first refer that such a name does not exist. This is something that is very, very important. And when you're trying to describe a new species, again, this is very important when you're trying to describe a new species, demonstration that a particular culture that it represents is already existing. That is a holotype. Holotype is already existing is what you have to demonstrate. So you have to describe that you have to tell what is the phenotypic. So you have seen if you have read about the Kanningamala Arunloke article, and if you have read about the Madurela Fahali articles, when these articles were first published, so these articles actually are describing how this particular species is phenotypically different, and where genotypic application of or variation from other species has been demonstrated. Then multiple data types are being mentioned in those articles, and there is a clear delineation of the species based on what are the phenotypic characteristics, what are the genotypic characteristics, what DNA barcodes were used to facilitate for rapid identification. So all these things are something which are written in detail when you're writing or describing a new species. So that is very important. So you have to write about what are the number of collections of these new species you have got, and there is some, was there any evidence of this new species earlier? Then what are the species concepts? So you have to stick to the definition of the species concept, and you have to tell from the geographical point of view, from which part it is important, from which area it has come. So you have to publish the DNA barcode sequences. This is very important. So it is recommended that wherever possible, the DNA barcode sequences have to be provided in a public repository. Minimally, at least, you may not provide it for isotypes or epitypes, but it is very important to provide for a holotype. So this is very important because it is the first one when you're trying to give it to one culture collection. So a public repository specimen has to be created, and this is very important, and it should be given the number. At the same time, you have to write about what are the sequences and what is the barcode loci, what has been used. Then compliance with the convention on biological diversity, and there are several protocols which you have to follow. So there are several new combinations, names at new rank, and replacement of the old names. So these are purely based on the typification. So holotype should be always selected if you are trying to give a new name. So a type serves as a standard for future comparative analysis. So if you are trying to do a comparative analysis, you should see that how many holotypes are there, how many isotypes are there, how many epitypes are there. So here the first thing that you're going to select is the holotype if you want to compare. So holotype should be selected where possible from a collection with adequate material for generating DNA barcodes and for designating additional isotypes. When original type material of previously published names is poor or ambiguous, so you have a new combination because you've chosen from various sites, so that becomes your epitype. So these are some of the important guidelines when you're trying to look into the writing of these new names. You have to check what is there and what is important. So describing about the diagnosis and the description of how it was diagnosed and how it was, this actually the code does not recommend nowadays with so stringently that publication of both the diagnosis as well as description is mandatory. So a diagnosis is a short statement that delineates the new taxon from similarly described taxon. So a diagnosis should aim to provide actually a statement that is, it tells you about the key diagnostic parameters. So nowadays the code does not mention it with such a significance that you need to publish all the parameters of this diagnosis. But still many of the articles, when they are describing new taxa, they do have this diagnosis, that what short statement of diagnosis they do write, which is very important. Then there are some things called as the description. Then you are proceeding further to describe. Then to further explain on these phenotypic or genotypic parameters, you add some pictures or you add some images or illustrations. So these images or illustrations have to be very clear, and they have usually used for morphological phenotypic features, and it is strongly recommended to support the descriptions. So authors should provide high quality images of new species. So you definitely should read those articles where there have been publications of the new species, and you will see that they are taking efforts to differentiate it from the existing ones, and they will tell you about the phenotypic characteristics. Each and every characteristic is very clearly tied. It is like on the culture plate, how this species looks from the other species. If you do a LPCB or you're looking under the microscope, how it appears, how the spores are looking, how the hyphae are looking, so there are each and every small detail, it is highlighted because you're trying to explain Then there are the barcode methods, how you're trying to differentiate, on what basis you have tried to differentiate it as a new species. So all these things are very important when you're trying to write about the new species. Notes are also again further which expand. So if you look at the articles, so I'll request everyone to look at those articles where there have been new names. You will try to see that all these aspects have been covered when people have been writing. So species author citations is also very important. The author's names for a new taxon also follow the naming conventions. So the names of authors of fungal taxa follow standard form, typically abbreviations, a complete list of which can be found on the Index Fungorum. So you write a author's name also with that. So that is again very important. You will see in most of the publications, there are authors' names also. There is a short form or citations that you will choose. And then you have got to choose only the online form of publication. That is very important. You have to use all your molecular data for publication. So in that molecular data, you have to write about the number of sequenced representatives and multiple sequenced representatives. If you had for a new taxon from different collections or isolates, you have to include that in your analysis. The number of sequenced loci and the sampling strategy for phylogenetic analysis. What was your strategy for sampling? This is again very important. So they want an explicit statement about taxon selection or sampling strategy. So these things are very important. Then data accessibility. So physical data, it is not mandated by the code that you have to keep a physical data, but you need to have the, it may not be physical data that they isolate or everything you have, but yes, biorepositories, recognized biorepositories, they should have this, all these repositories, they should have those species where it can be accessed again and again. And so this is what you understand by the physical data. So very important task for the individuals who want to write about the new species is you want to start with the repository, because unless and until you don't put it in a repository, it doesn't become your holotype. So selecting a repository is very important. So whenever possible, ensure that the repository is the one in which access to materials via loan or other means is readily obtained or freely available. So that is very, very important. You have very active culture collections are there. So you have to choose those culture collections which are active. Some culture collections are not active. So you have to choose those culture collections where living actively propagated material, they can preserve it very well, and they give you a holotype number. And this must be always preserved in metabolically active state, and preferably by cryopreservation or by lyophilization. So these are very, very important. They are stocked in the form of cryopreservation or in the form of the lyophilization. So these are some things that are very important. This is about the code decoded. You are writing, doing an effective publication, a valid publication and legitimate publication. So these are some things which you should have, or it is very important for all of us to understand. Then you have to submit data including both specimen metadata and molecular sequences to the public online repositories. So that is known as digital data what you are going to submit. So these things are again very, very important. And further, after that digital data, that primary you have submitted, there will be sequence submissions that are done. So these things are very important. What was the blast? NCBI qualifiers you are giving. So most commonly used modifiers, they recommend public biodiversity sequence records. Like you want, it should mention this particular biodiversity record, it should mention with whom it was collected, name of the person who collected the sample, collection date, country, then culture collection, whether it was from which culture collection, which institute is maintaining that, host, who is the host. So fully verified binomials have to be used for the host, isolation source, notes, specimen vouchers, strain, tissue type, and type material. So these things are very important in the code decoding. So there are a checklist for the publication of the new species, which is very important that we need to understand. So before I stop here, I would like to discuss something about the checklist that we are going to have. So I will also tell you the reference of the article which you should refer, that is ME et al. IMA fungus. And this article is very important. It tells you about all these descriptions in a lot of detail. So there are some checklists for publishing journal selection. It should be peer-reviewed journal and meets requirement for effective. That is, as we have discussed about the effective publication by Turland. and that is the code decoded. So we have understood what is effective publication. So it meets all the requirements. If some journal that you have chosen, it is peer reviewed, but it is not online or there are some issues, so you should not choose such a journal. Then taxonomy, it should ensure that the name is unique and it is unlikely to be confused. There are no cross-code homonyms, and ensure all taxon authors are involved. So you want all authors to be involved in the taxonomy determination, and name is indicated as a new species or new combination. So these are some things which are very, very important, and you have to have a repository and the repository number that you have to mention, and include short diagnosis in English or Latin. And these things are important. Then type material designation, holotype, it has to be correctly designated. Holotype voucher, additional isotype if somewhere else you have kept as an isotype, or if there is a paratype, or you have got previous culture types that you already exist. So all those things you have to give. Species description and notes is very important. Manuscripts, like what your keywords and all these things, manuscript writing. So all these aspects of manuscript writing, you have to write it very, very clearly. And data deposition is very important. And post publication also, you should ensure that all data repositories are updated. If suppose after publication there is something new you are adding to that species which has been described by you, it has to be further added in PDF file only. And ensure type material is correctly indicated in the NCBI taxonomy. So these are some of the fundamentals what you have to read. If you wish, I can share with you this article. I'll just show you. We have. So we will start discussing about that. I'll share you with you the information. Followed by that, I will share with you that Kaningamela Arunloke article. These two are very important when we're discussing about the novel species. So this is the article which I was mentioning. So I'd like to share with you. So I request all of you to read about this. This is AIIMS et al. IMA Fungus 2021, and it is open access. And how to publish a new fungal species or name version 3. So you are getting all the details, whatever I discussed here. You can click on this. You can go on the chapter F if you want. You can go on the Terland 2019, the code decoded, what are various instructions. So it gives you everything. Then how is the registration nomenclature and what are these different repositories? You can just enter and explore these repositories. So these are some things which are very important. I discussed to you about the holotype, isotype, epitype. So all these are there. Effective publication, valid and legitimate publication. Then significance of taxonomy and describing new species, how you have to write, what all characteristics you have to mention for new species. So these are some of the important aspects that you have to cover when you are discussing about a new species. So they are all written very clearly. So if you wish, you can go through it and read it carefully. So these are some of the important aspects, what you write, most commonly used modifiers that has been mentioned. Then you have got the checklist for publishing new species. So there's a very beautiful checklist here, which you can see. So everything is mentioned. They say that the required actions are in orange and desirable actions are in blue. So it will again give you a lot of clue on that. So there are new species of sporulating fungi, which have been mentioned. So it is entered here. There are new species of cultured fungi, including yeast, which you can follow from here. And we can click it and observe what are these new fungi which we can see. So there is a whole lot of list of different fungus, new combinations that have been submitted. So this is how I wanted to explain to you regarding the significance of all this. So this article is very good if you all can just go through it once. So we'll discuss that another article through which we'll try to understand how it is actually important. So this is the one which we are actually seeing it. So we can have, we'll just zoom it once. So this was the new species, Kaningamela Arunloke. So a new species of causing disease in an immunocompetent individual. So this is very, very important how we can understand. So it gives the description of all authors. And it tells you about the introduction is there and how they have done all the material methods, phenotypic characterization, how it was done, Then molecular characterization in entire detail. This is important. Phylogenetic tree construction and whole genome sequencing. So they have all the details mentioned with them. So all the repositories and every code has been given to it. So this is how you progress. So phylogenetic code is given. NCBI accession numbers have also been given for the sequences. So, and the clinical description of the patient, they have mentioned it, phenotypic and the morphologies have been mentioned with a lot of clarity. This is phylogenetic tree, and this tree, they have tried to describe the association and the difference between other species. And regarding the morphological aspect and taxonomy, again, they have mentioned that it has been deposited with the Mycobank, and the Mycobank number is there with authors' names with whom it is submitted. So this is again very important. So they are describing us that what type it is, isolated from sinonasal tissue of patient with chronic mucormycosis. So this is a holotype that they're saying that it is dried culture at NCCPF Chandigarh. So just now, whatever I told you, it is just, you can go through this as an article and you can compare between that article and etymology, like it is the terminology, the species epithet they're writing, like it is Arun Loke, how it got the species epithet. So in honor of Arun Lok Chakravarti, and colonies description, again it starts. So this is how you're going to progress when you're trying to read a new species. So they are saying that it is deposited with the NCCPF. So you read that article, you read some of the new species that have been discovered, and you will understand that this is so important. In present scenario, it is much more important, where we have lots of new species coming up. So they have very beautifully tried to describe about the diagnostic clinically also, if there is any difference or similarities, then showing phenotypic characteristics that has been tried to be described very clearly. Then phylogenetic delineation of Cunningham species based on what. So all these things have been taxonomical classification into which it fits, complete barcoding. So these things are very important. And then there is a supplementary material they are saying. So this was actually available online. So this is how it has to follow. So it fulfilled the criteria of the effective publication. Then you have got the all the aspects of it, valid, which is valid publication, legitimate publication. So these are some of the aspects which need to be followed. So I hope I would have done justice to this topic, because it is even challenging for all of us to know, because this is something that we don't do routinely, and but these things do come us in front of us. And when we are reading these papers, we have to understand, and we have to very critically, and we have to see them very carefully, so that we know, we understand the science behind it, we understand the hard work behind it, we have to know how it was done and what all things have been done. And once this species is written, so we have to know how it happened. So there are a lot of such articles which come up. So we have to be very much careful in reading and interpreting those articles. And they are very helpful, because they always encourage us, if there is such a thing, how we are going to write. So this is something very different kind of publication. So writing about new species is a very different kind of publication from what you're writing routinely about any epidemiological study, case control study, or any other study, intervention or trial study. So this again is equally challenging, like any other publication. So this was my final topic that I wanted to share with you all, because while reading through it and with each aspect I was going through it, what is the significance of this component, what is the significance of each and every word is important when you're reading it. So you need to read it with lot of care. So every word where the sequences have been kept, all these things are very important. So we need to read it again, so that we refer to that article and come back and we understand what all things we have understood and what we have not. So all the best to all of you. So this was what I have discussed. So I stop here, and the session for mycology almost it ends. So today and tomorrow it is mostly about the biostatistics. So I stop here and I stop sharing the article.